All right. Hi, everybody. So we are going to run through a quick review of chapters 7 and 8, which are going to be the content of the third quiz. This is going to be the last quiz before the midterm. So we're going to just look at the content that's included. Now, to remind you guys about the expectations of quizzes here in the world of microbiology, they're going to be 40 questions, each of those questions worth 1.25 points, which means it's a total of 50 points. In order to give you adequate time, you get 60 minutes or a minute and a half per question as your maximum time available. Just a friendly reminder, guys, the questions are presented one at a time, which is going to mirror a little more similarly some of your upcoming classes. This class, we're going to do that. You guys are going to need to utilize Respondus. And then when you return, we're going to go ahead and crack into chapter 9. So we'll be doing microbial genetics upon return. All right, guys, so first thing we're going to do is go through the major insights into chapter 7. Chapter 7 is the nutrition chapter. So it really is very basic in terms of the major content. We defined nutrition. We explained the difference between macro and micronutrients. We talked about types of trophs, so determining the different preferred food sources for and energy sources for different different types of organisms. We discussed the features of membrane transport, which is really the way that the microbes will be eating. Then we talked about the required conditions for growth. We finally went through and looked at how growth can be viewed on a growth curve and talked about techniques to actually measure growth of a population. So from here, guys, let's go ahead and talk about the details we covered in this chapter. The first thing we really learned about was what is a nutrient. Nutrients are defined as molecules that are essential or required for growth. Essential nutrients are things that can't be produced inside an organism, so they have to take them in from outside. Now, that actually falls into categories of those essential nutrients that are either larger in size and need to be taken in larger amounts, macronutrients, which these guys are going to be what you think of as your big organic chemicals. So going back to chapter 2, you were able to go through and look at those molecules that have carbon, hydrogen, and then a variety of the other different types of living elements, like you see Chopkins as your friendly reminder. These guys are the ones that are going to give your cells the energy they need to sustain themselves. So starting in order in terms of how that energy is achieved, carbohydrates will give your cells carbon, hydrogen, and oxygen in a 1-to-1 ratio. Lipids or fats are going to give it just a lot of carbon, hydrogen, and oxygen, and actually are the second source of energy. So they're a high energy molecule. Then proteins can give your cells carbon, hydrogen, oxygen, nitrogen, and sulfur. But it turns out that nucleic acids can also give your cells carbon, hydrogen, oxygen, a lot of phosphorus, and some nitrogen as well. Now, those are the macronutrients that give the most energy, but cells do not survive without small amounts or trace amounts of micronutrients. And really, when we think about micronutrients, these are going to be like the vitamins and minerals. And there's a list of a variety of different minerals. I'm not having you guys memorize individual sources for any of those minerals. Mainly, guys, it turns out the main type of element we spent the most time looking at where you get it is carbon. So carbon sources, you guys want to remember that they are every organism on this planet is made up of different chemicals that have really been recycled from different elements on Earth. So, and in the terms of the fact that energy is neither created nor destroyed, matter is neither created nor destroyed as well. So the carbon that is incorporated in living systems of today is the same carbon that's been on our planet since the time of the dinosaurs. So it's important to remember that it just depends on where the source of carbon is. In terms of the source of carbon for phototrophs, they're going to be utilizing the energy from the sun and performing photosynthesis, incorporating carbon dioxide for their different types of carbon molecules, whereas a chemotroph is going to take its nutrients from other chemicals. And so this is going to be an important way to define that. Now, going into it too, we know that we have heterotrophs, where the materials are taken in from an outside source, and autotrophs, where they are self-feeders. So to kind of break down those different trophs, I want you to remember that you are a heterotroph, which means you get your chemicals by consuming them from other sources. And so other examples of chemoheterotrophs in the world of microbiology include saprobes, where they get their foods from dissolving enzymes outside the body, and that's how they decompose, and then they just literally suck up that decomposed material through their plasma membranes. Or there are also parasites, and what they do for their food sources is they utilize a host and then take the resources from that host directly. Either way, that principle of carbon metabolism is the only major chemical I had you guys review in terms of what you needed for this chapter. But moving forward, we have to remember what causes a microbe to eat. And what causes a microbe to eat is actually absorption through the membrane. So microbial eating is membrane transport. There are two key types of membrane transport: passive and active transport. And the reason why membrane transport is something we discuss in detail is to help you remember the different features of those two types. What we need to do specifically to ensure that membranes molecules can cross a membrane is to consider how that membrane is structured. With a phospholipid bilayer, the membrane has molecules that can freely cross it because they are lipid soluble or nonpolar. Examples of things that can freely cross a membrane include respiratory gases like oxygen or carbon dioxide, or even other types of waxes or fats can cross membranes. Things that need carriers or a little bit of help to get across include things like water, which uses pores called aquaporins, sugars like glucose, proteins and amino acids are typically charged and have a hard time getting across a membrane, and then a bunch of other charged particles that, when dissolved in solution, can even conduct electricity, called electrolytes. So all of those minerals need a way to get across membranes. So getting across a membrane is important, and membranes keep some stuff out and let other stuffs in, but in order to cross a membrane without any energy, that's called passive transport. The type of method of passive transport is going to be moving down a concentration gradient, crossing without energy. So great example of something that passes through the membrane without an issue, always going down its concentration gradient, will be those respiratory gases. Another type of passive transport that's facilitated with the help of that pore in the membrane is water. Now remember, water is always going to move in the direction of the salt or the solute. And so as water moves, it's trying to cause the conditions on the inside of the cell to be equal to the conditions on the outside of the cell. By doing so, water is trying to achieve something known as an isotonic solution, where the conditions are the same concentration inside versus outside. And if that's possible, the water then ends up with no net movement between the inside to the outside. However, if there is a hypotonic solution around a cell, the cell will swell up, and if it doesn't have a cell wall or any other protective cell envelope, it will actually swell to the point where it bursts. So a helpful way to remember that is hypotonic has the letter O in the word itself, and that's the kind of shape the cell takes right before it explodes. So the big O is like an exploding cell. Hypertonic solutions that surround cells, these would be really salty solutions. These guys will actually cause the cells to shrivel. As the water leaves the cell, it will try to make the environment around it equal, and so the cells will shrivel. And again, the cell wall on microbes can actually keep them, act like a capsule around them, helping them to survive until conditions become closer to isotonic again. So these principles of passive diffusion don't use any energy, but occasionally a microbe is going to invest the energy necessary to get certain things into or out of its cell. We have embedded in the membrane certain types of pumps, like a famous sodium-potassium pump has been previously discussed, but also another key way that microbes will eat in large amounts, they'll actually reach out and grab nearby food particles or even other nearby organisms. And that'll be in the form of phagocytosis, kind of like taking a big bite. The other option is pinocytosis, where they take a small pinch of nutrients from around them. Okay, membrane transport got a lot of strategies or techniques, and this is a concept you've learned previously, so I encourage you to use a lot of video help to reinforce this. Okay, so when a microbe is growing, what is actually happening is the microbe is growing in population size. And in order to make that happen, prokaryotes themselves undergo a special type of division known as binary fission. So binary fission looks like mitosis, but is just a little bit easier for bacteria because they don't have to go through as many complex steps as mitosis. Microbes will begin to grow and they have dedicated growth requirements. So of the first type of growth requirements, temperature is something that they can have pretty specific requirements on. For every bacteria, they have a set of cardinal temperatures, basically the range of temperatures that they can survive within. So for that, there's a minimal temperature for survival. If you go below that, they will oftentimes go really dormant. Occasionally, they'll freeze to death. On the other side of it, though, they also have a maximal temperature. And this is the highest temperature before you begin to start denaturing their proteins. So if you go higher than that temperature, they won't survive most often because their proteins internally will start to fall apart. After you get past this, though, guys, you have an optimal temperature. And an optimal temperature is going to be the ideal temperature that they will grow at. It's their favorite temperature for survival. So to do this and to give you a little bit more foundation on this, you have actual microbes that are within a range of different survival temperatures. Psychrophiles or psychrotrophs themselves like cold temperatures. They'll be the ones that are responsible for freezing or food spoilage that can happen inside a refrigerator or a freezer. If you have mesophiles, these are definitely your most interesting ones because these guys are going to be able to grow in relatively similar conditions to the conditions that you prefer. So they'll grow in conditions preferring usually around 20 to 40 degrees Celsius as their optimal ranges. They can go down to as low as near freezing and up higher than body temperature, but they really like temperatures that you really surround yourself in. And for many of them, their ideal optimal growth temperature is right around human body temperature. So mesophiles are famous for being those type of microbes that make up the majority of human pathogens. The last type to go for and to understand are ones that can actually survive really hot conditions. Conditions that will famously denature others can be actually ones that these guys can thrive through. So thermophiles or extreme thermophiles can survive in conditions that would pretty much melt or burn other microbes. So these guys are going to be on the other extreme end of the spectrum. Now, temperature is one growth requirement, but another type of growth requirement is definitely what type of respiratory gases are present. Aerobes like oxygen, and there are aerobes that are obligate aerobes. They can't live without oxygen, kind of like you and me. There are facultative anaerobes. These guys have the ability to switch back and forth. Depending upon the conditions. There are ones known as microaerophiles. They do like a little bit of oxygen. So these guys are like ones we'll find within about an inch of the top soil layer. Then there are other types of anaerobes. Anaerobes don't like oxygen. Now, obligate anaerobes will literally die if oxygen is around. Aerotolerant microorganisms, these guys will kind of put up with whatever. So if oxygen is there, they'll grow. If oxygen isn't there, they'll grow. And they don't really have an issue with any air. Air doesn't play a role in their growth cycles very much. Now, there's a small type of microbes that really actually will utilize CO2 for those survival, and those are called capnophiles. All right, another set of growth conditions can be things like pH or salt. So most microbes really like a pH somewhere around 6.5 to 7.5, which again, I'll point out is pretty similar to human pH. These ones that can take on the lower end of the spectrum that like acids are known as acidophiles. There are even a few that like to live on the high end of the pH scale, and those are known as alkalinophiles, so they like bases. The ones that are salt-loving microbes are called halophiles, and they like the salt so much that they can survive any of those changes in osmosis that would normally shrivel a cell. They thrive within that environment. There are other things like barophiles and radiophiles, but you'll notice that those aren't on your summary slide because they aren't concepts that I ask you guys to test on. Now, the final condition that really will impact a microbe's growth is the association between different organisms. Some organisms will need each other. That's known as a symbiotic relationship, and other organisms may have a take it or leave it type of approach to their relationships. That's known as a non-symbiotic relationship. Under symbiotic relationship, there are three types. The first one is mutualism. In this scenario, the organisms both benefit from having a relationship. Commensalism will be when one organism benefits, the other's not harmed, so it's a good relationship for both of those involved. The third one, though, is parasitism, and in this scenario, one will benefit and the other is harmed, so this is not a beneficial relationship for both parties. On the other side, there can be a set of non-symbiotic relationships that happen, and occasionally there's an overall sense of cooperation that's called synergism. This happens in things like biofilms. In fact, there are little tiny chemical signatures sent out by microorganisms that will actually encourage the growth of biofilms. Once the biofilm grows too big, though, the same molecules that drew organisms into the biofilm can also help them to set up residence in a different location. And so biofilms are able to sense the size by a phenomenon known as quorum sensing. Finally, non-symbiotic relationships can happen in which one microorganism actually can outcompete the other. And this can be an example of antagonism. Now, microbial antagonism is one of the key things I want students to be familiar of, because it is the reason why your body has healthy bacteria, or what we call your microflora. If you have healthy bacteria, they can reduce the likelihood of unhealthy microbes for growing in your system. And so an example we talked about under this particular aspect was the impact of antibiotic use. If you knock down your healthy bacteria with the use of antibiotics, that can actually alter or affect someone's health. Okay, guys, now talking about growth is only part of the deal. We know that if we're analyzing population growth, what we're really looking at is cells dividing. And the amount of time it takes a cell to go through that division, binary fission, is known as the doubling time or the generation time. For every microorganism, they have a generation time that can be measured, and you can optimize that by creating optimal growth conditions. Now, what we can do is we can look at the population size of microorganisms with the help of a growth curve, and I have an example of one added here. The growth curve starts with a lag phase where there's no visible signs of growth, and the numbers are just too low at that point, leading to a log phase, or commonly called the exponential phase, where the numbers really begin to become measurable and the growth is in high amounts. The next phase happens when the resources start to become limited. It's called the stationary phase. And in this phase, the numbers are leveling out. So the amount of new cells being born or being divided is equal to the amount of cells that start to die. Now, growth curves will continue onward, and once the resources are utilized, the fourth phase, known as the death phase, is going to occur. And this is when the cells are now dying at a rate greater than they are surviving. Now, what's amazing is usually a few microbes find a way to survive in the end, which allows this population to actually carry on for the future. And growth curves are things that we use to measure all of these events. And it's pretty important. We went through and explained the impact of misuse of antibiotics associated with this. So this is just a simple way to visualize and help you understand why proper consumption of antibiotics has been an important thing to emphasize. Now, talking about that measured growth, we know we've got that growth curve to look at it on a scale, but if we wanted to just grab a sample of microbes, one of the mechanisms we might do is we might try to grow those microbes and count using a little bit of math to calculate the amount of colonies on a plate can help us determine exactly how many microbes are in a solution. Another method, we could actually take some microbes and put them onto a slide and literally count 1, 2, 3, 4. So there's a couple of direct methods. Two of the less direct methods include measuring the turbidity. That's the cloudiness of microbes in a solution. And so it measures the density of microbes and uses the amount of light that makes it through as a way of determining how many microbes are in a solution. And then the last one that we talked about a little bit more, we showed off how some microbes can be passed through a very thin capillary, and each individual cell can be counted almost like a counter at a gate. So each individual cell can be calculated so you can figure out how many microbes are in a population. All right, guys, this completes chapter 7, which just leaves us to look into chapter 8 itself. And chapter 8 is definitely one of the more challenging chapters, so we'll start by doing some chapter insights. If you will recall that this chapter is the metabolism chapter. So in the beginning, we took some time to define metabolism. We addressed a very simplified model. It looked like a Nike swoosh, and we'll show you that in a moment. And then we looked at how enzymes help to facilitate the various reactions of metabolism. We went through looking at the function of them, the structure of them with apo, cofactor, coenzymes, and holoenzymes. We learned and talked about how substrates bind to active sites, and then addressed the different types of enzymes and the availability. From there, we also went into looking at the key types of metabolism. So we compared catabolic reactions for carbohydrates with and without oxygen. So we did aerobic respiration, and we did mostly a focus on fermentation form of anaerobic respiration. Before we got there, though, we did address the fact that you have to utilize either substrate or oxidative phosphorylation. And oxidative phosphorylation reminds us of the fact that there are reactions in the body where electrons are moved. Leo the lion says grr. We reminded ourselves the structure of ATP. Then we went through the steps of the pathways, but mostly focused on how to take away yield from each of those. So glycolysis yield, remember which pathways use those gases. We then looked at the total yield. Along the way, we collected some taxicabs, NADH and FADH2, and we compared how prokaryotes have just a single cytoplasmic membrane with a low surface area versus eukaryotes that have mitochondria with a high surface area, and how that affected efficiency. Going through this, the metabolism of microbes is defined as the combination of anabolism plus catabolism. And previously we learned about anabolic reactions and catabolic reactions. And so you've also had a chance to learn about synthesis or decomposition reactions. Helpful ways to remember this are to try to remember whether the reactants are greater in size than the products. And so catabolic reactions, the reactants themselves are going to be greater inside than the product, which means a bunch of bonds are going to be broken, and therefore ATP is going to be released. Anabolic reactions actually have the reactants smaller than the products, which means you're building some stuff. And in order to build that, these guys are going to require ATP. So what happens in nature is we couple anabolic and catabolic reactions to make sure we have the energy necessary to do both. And in order to get there, guys, catabolic reactions happen on part of our Nike swoosh, and anabolic reactions happen on the other part of our Nike swoosh. I'm going to pull the Nike swoosh up. I make it a joke. It's not really a Nike swoosh, but it's got that check mark type structure to help you see the events of these reactions. So on the catabolic side of things, you go from larger molecules down to precursors. What you have are the events of glycolysis, the Krebs cycle, and the electron transport chain. Or, of course, if oxygen is not present, fermentation. These events of catabolism take glucose from its big 6-carbon sugar and begin the production and breakdown to the precursors like acetyl-CoA or even pyruvate. Now, these same precursors can be used to go anabolically and build up to other molecules. And when it comes to becoming the building blocks, those will be things like amino acids, monosaccharides, basic nucleotides, or even fatty acid chains. All right, amino acids are the building blocks of proteins. So an example of that would be enzymes. Monosaccharides or simple sugars become polysaccharides. So some examples in microbes include chitin or even the peptidoglycan or lipopolysaccharides. Nucleotides like adenosine, thymine, guanine, cytosine, and uracil become the molecules of DNA or RNA. And fatty acids themselves can become more complex fats. They can form phospholipids in the membranes. They can become waxes like on the cell envelopes or even part of those lipopolysaccharides. These guys are small. These are getting larger, so the swish is moving upward. Combine a bunch of these together in just the right way and a cell can be made. And here's where we can compare the difference between prokaryotes and eukaryote cells. Okay, guys. Now, in order to really go into metabolism, we had to first look at the major impact of the metabolism itself. And so we look at what helps to make reactions happen faster and more relevant. So the enzymes are the protein-based molecules that speed the rate of a reaction. And when something speeds the rate of the reaction, it's going to be also termed a catalyst. Now, some features of these catalysts are that they lower the energy of activation. They basically take a huge mountain of a reaction and turn it into a smaller speed bump. And as they lower the energy of activation, their function is to help make that reaction occur, but they aren't actually used up during that reaction. They're just making the reaction happen at their active site. All enzymes end in ACE at the end of the name. So it lets you know you're talking about an enzyme if you hear the ending with the name ACE. There are several types of enzymes involved in anabolism and catabolism, but I don't have you memorizing those classes or groups. Enzymes themselves can be just the protein, which is called an apoenzyme, and the structure has to fold from a primary to a secondary, secondary to a tertiary, and then tertiary to a quaternary structure. At this point, the apoenzyme is just not folded correctly without the help of the cofactors, which might be metals or things you know of as minerals like magnesium or iron, for example, or coenzymes, which are typically organic molecules like different types of vitamins, or even you've heard about coenzyme Q10, for example. These little factors help the enzyme to fold properly and create the necessary active site for the substrates to bind. Together, you take these components, the apoenzyme plus the various factors, and you get a holoenzyme. And a holoenzyme is really that ready-to-go molecule where the substrate can bind to the active site, and that active site is the location where the reaction occurs. Enzymes carry out different types of reactions, and with the only ones that we went into was explaining how a group of enzymes called oxidoreductases might carry out oxidation reduction reactions. So if a molecule loses an electron, it becomes more positive, and the term for that is to say that it was oxidized. Lose electron oxidation. If that electron is added to a different molecule nearby and it gains an electron, it actually becomes more negative or gets reduced. So reduction is gain electron reduction. Enzymes that exist inside microbes, there's groups of them that stay inside the cell, endoenzymes, or there's some that are actually secreted outside the cell, exoenzymes. A great example of those would be how those saprobes digest their food sources or decompose. Also, we know that enzymes are going to be regulated pretty strictly. Now, some cells will make enzymes and keep them produced constantly. That's called a constitutive enzyme. But other cells, because they're trying to conserve energy, are going to regulate their enzymes. They will turn them on or induce them when the conditions are favorable and the substrate is present. And then once the substrate is used up or goes away, they will turn them off or repress them. And this is a mechanism to ensure that microbe can survive without wasting excess energy. Now, enzymes themselves have those specific shapes that carry out their function. If you change the shape them, or if you block the active site, you can actually block the enzyme activity. So one type of enzyme blocking is called competition, and competitive inhibition actually blocks at the active site. This is one mechanism for drug development to block specific microbes' function. Another thing that can be done is non-competitive inhibition, and this one will actually bind to a different site, but it will change the shape just enough so that that enzyme is non-functional. And these different types of competitors are things that can work to kill a microbe, or even things that are used by microbes to perform microbial antagonism. Another strategy for breaking apart an enzyme structure might even be just to denature that enzyme itself. And that can be done with high temperatures, or even things like low pH is really great at doing that. Now, the process of metabolism has to use enzymes, and enzymes are never used up in that reaction, but the enzymes will just keep popping out these reactions very quickly. Well, some of the most important types of enzymes we'll see going through this process, but we don't actually have you guys memorizing any of the specific enzymes involved in metabolism. Now, hold on tight, guys, because in chapter 9, we will do a review of enzymes for chapter 9. All right, guys. Now, one important thing to be aware of is that in order for you to go through this metabolism, a series of exergonic reactions where energy is released, or endergonic reactions where energy is required, you need to have ATP as the cash currency of the cells. ATP molecules have high energy bonds, and you guys can remember the structure of that. It's an adenosine with 3 phosphates hanging off the end of it. Because those high energy bonds exist, you can actually take the phosphate group, not seen as the PI here, bounce it onto ADP, adenosine diphosphate, and convert it to ATP. And in nature, ATP can lose a phosphate, go back to ADP, gain a phosphate, go back to ATP, and go back and forth and back. And so in nature, this is the strategy to utilizing the ATP in those endergonic reactions, and then releasing ATP in those exergonic reactions. In the meantime, that phosphate has to find somewhere to go. So there are 2 types of phosphorylation events. The first one is called substrate level phosphorylation. This is when a different molecule will kind of hold on to the phosphate and then hand it back as needed. So I equivalated that to kind of holding someone's bags. So a substrate holds onto the phosphate and then gives it back to ATP when necessary. Oxidative phosphorylation, this is done by those really cool looking taxicabs of the cell, the NADH and the FADH2. And these guys will help hold on to the energy necessary to transfer those phosphates. So oxidative phosphorylation is another mechanism. And in fact, this one is going to give us the most energy during the electron transport chain. Okay, guys, now photosynthesis is discussed at multiple points throughout this chapter, and it's really cool to know because it generates oxygen for breathing, but the steps of photosynthesis and the nuances are not things that are required for your quiz. All right, so let's go ahead and break it down. And this is the big summary slide. To avoid redundancy, it's all on one slide for you guys, but I want you to remember that this is breaking down a bunch of different steps associated with this. Now, this is kind of typed out. Of course, you guys can utilize more of a flow chart mechanism, and you might like to see them put together in that way. I sometimes even use a table setup to see these numbers calculated. So glucose is a 6-carbon sugar. It has 6 carbons, 12 hydrogens, and 6 oxygens, and that's the structure of glucose. So it's a basic carbohydrate. Glucose is the preferred energy molecule for cells' energy. Well, once that is present within a cell, the cell can begin to break it down in a process known as glycolysis, where it converts it into 2 pyruvates, which are 3-carbon sugars. Along the way, a few ATP are generated, and a few of these electron taxicabs are generated. After glycolysis, there's a small intermediate step in which those 2 pyruvates are actually converted to a molecule known as acetyl-CoA. Now, this is only a 2-carbon sugar, so 2 carbon dioxides are actually released during this intermediate step, and you get a couple more of those taxicabs. Now, it's going to be these acetyl-CoA molecules that are going to begin their adventure down into the further pathway of the Krebs cycle, sometimes called the citric acid cycle. The two acetyl-CoAs, each one goes through a single rotation of the cycle, but each of these acetyl-CoAs goes through the cycle, and as a consequence, you generate 4 CO2s, 2 more ATPs, 2 NADHs, and 2 FADH2s. So we've got a bunch of taxicabs and in total 4 ATPs at this point in the cycle. At the end of the citric acid cycle, you have completely broken down the entire carbon structure. 6 CO2 molecules have been created, and so the cell has in essence breathed out those CO2s. Now you've produced 4 ATPs to this point, but what's really going to yield the most energy going into the electron transport chain are these electron taxicabs. For every NADH you have, a cell can make approximately 3 molecules of ATP in that electron transport chain. For every FADH2, it can make approximately 2. So since there were 10 NADHs and 2 FADH2s, if you go through the electron transport chain, you need a little bit of oxygen right here to serve as the proton receiver, prevents the cells from getting too acidic. And as a consequence, the 10 NADHs produce 30 ATPs. The 2 FADH2s make 4 ATPs, and then the 6 oxygens combine with all those extra hydrogens and make 6 waters. Okay, the steps that you see here, glycolysis, intermediate step, Krebs cycle, and the outline of the events of the electron transport chain yield one key summary reaction. And remember that the numbers on ATP will be shown to vary because inside different cells, sometimes the efficiency goes up or down. Glucose starts us all off, and this is why sugar is the primary source of energy in cells. This glucose goes through those processes of glycolysis, through Krebs cycle, through the electron transport chain, and it utilizes 6 oxygens in order to breathe out 6 CO2s and make 6 more waters. And these 6 waters over here are going to just contribute to the water produced inside your cells. But the main reason for all of that hard work was to yield somewhere in the range of 32 all the way up to 40 ATPs. And that's a pretty decent yield for the cells to survive off of. This is what's happening in the case of oxygen metabolism, and this only happens because oxygen is present. If oxygen is not present, there's a different set of metabolism we'll go through in a moment. But I just want to review one final takeaway from aerobic metabolism. Remember, this is happening in those heterotrophs that are getting carbon from their food. We know that glycolysis is what we call ubiquitous, which literally means everyone does it. So this glycolytic pathway is present in cells that breathe oxygen and cells that don't breathe oxygen. Glycolysis itself does not require oxygen, only the electron transport chain does. So it's important to remember that everybody can do glycolysis. Glucose is what enters glycolysis and serves as the first source of energy. Then remember that this will lead to this downstream pathway where ATP is generated. If prokaryotes are performing these processes, they only have the surface of their plasma membrane in order to do the electron transport chain. Their lower surface area means they generate slightly less ATP and they're less efficient. So they really have to conserve their nutrients. On the other side, eukaryotes have a bunch of mitochondria, and in tissues in your body, like your muscle or your nerve tissues, you might even throw a few extra mitochondria to really bump up the energy. Well, this gives such an increase in surface area that these eukaryotes are much more efficient at energy metabolism. And this is actually why eukaryotes why the endosymbiotic theory of eukaryotes is highly supported, because these mitochondria make it possible for the cells to be larger and to generate more energy. Now, the main thing to remember is when you're thinking about prokaryotes, though, that they have to focus on conservation. And for them, conservation is a key strategy for their survival. So they don't make anything that they don't need, and they don't continue to produce things that they aren't using. Now, we remember that breathing in the presence of oxygen is the most efficient. So aerobic is hands down the most efficient. Sadly, anaerobic metabolism starts by getting you through glycolysis, and now you've generated that pyruvate. Pyruvate itself is an intermediate molecule that can go onward to become a building block, like you saw earlier with the big Nike swoosh, or it can continue downward if oxygen is not present and go through different pathways of anaerobic metabolism and fermentation. Fermentation literally only has the 2 ATPs that were generated during glycolysis. And fermentation in itself continues metabolizing that pyruvate into either acids or alcohols. And fermentation along the way will also produce a bunch of CO2. So examples of acids could be things like lactic acid. Microbes can produce lactic acid, but even humans can do this, which is the reason for sore muscles and for for acid content in your muscle tissue. However, another category of fermenters can actually produce alcohols. So ethyl alcohol or ethanol is what makes different types of drinking alcohol like wine and beer and all the different spirits. It turns out too that also some of this fermentation is important for the production of cheese or the production of household items like vinegar. And sadly, it's also the reason for the disease progression of something like gas gangrene with a Clostridium infection. Okay, so fermentation is a low yield event, much less efficient, and not as ideal. So right off the bat, thinking back all the way to chapter 7, that lets you know that obligate anaerobes or microbes that are anaerobic, they just don't grow as efficiently as the ones that are aerobic. Now, the metabolism of breaking things down is here. At the very end of your chapter, there is a brief review of the building up. Remembering, though, that you already learned about the basics of the building up. Pyruvate or acetyl-CoA, those precursor molecules, can generate the necessary building blocks for amino acids, for glucose to become even larger polysaccharides, for fatty acids to become more complex fats, or even for nucleotides to become nucleic acids. There are two types of sugar metabolism: gluconeogenesis, when cells are desperate for energy, they can actually make their own glucose, or glycogenesis, when cells will have excess glucose, they can store a few more extra molecules in the form of glycogen. And so this is actually something humans will do to store extra energy in their tissue. Now, these are concepts that are covered again in the metabolism section of A&P. All right, guys, so that's all she wrote.